Do you notice that weird looking toe? Maybe that burning and stinging raw skin between your toes? Or that torturous itching on your scalp? That's probably due to this friend right here, Trichophytum rubrum. Parts of Trichophytum rubrum include the macrocandida. It's a multicellular spore that is spindle-shaped and acts as an infection propagules, while the microcondia is a unicellular spore that has a slender clavicate shape and also acts as an infectious papugules. A hypha is a long, branching structure of a fungus and is collectively called a mycelium. Trichophytum rubrum is classified within the kingdom Fungi, phylum Ascomycota, class Urothiomycetes, order Onygonales, and family Atherodermatites. Notably, members of the Onygonales are commonly keratin-philic. Trichophytum belongs to the dermatophyte group. Dermatophytes are a class of fungi that require keratin for growth. Some of its microbial relatives that also work in crime is the Trichophytum tansurans and Trichophytum interdigitales. However, Trichophytum rubrum is a little infamous around here, since 90% of chronic dermatophyte infections are caused by it. Additionally, they are anthropophilic, meaning they prefer or love human hosts. Trichophytum rubrum's interactions with the environment primarily revolve around its survival and transmission outside of a human host. Trichophytum rubrum can survive off the human body as a spore. It can contaminate surfaces in the environment, especially publicly shared areas such as swimming pools and shared personal items such as toenail clippers. Transmissions occur through human-to-human contact or contaminated objects or surfaces. The infection process begins with the release of condylial adhesion that binds to the keratin. The condylia germinates into the hyphae. The hyphae invade keratin cells by secreting keratinolytic proteases. Keratinases belong to the class of proteases that are able to degrade keratins into amino acids. The amino acids is a source of fungal growth and metabolism. Trichophytum rubrum primarily has pathological impacts as a human pathogen. These infections include tinea pedis, athlete's foot, onychomycosis, nail fungus, ringworms, dermatophytosis, and more. In a chronic form, Trichophytum rubrum can invade the deeper levels of the epidermis and prolong symptoms such as ichthyosis vulgaris, fish scales, or scaly skin, plaques, and lesions. Normal foot skin is colonized by harmless commensal microorganisms that include bacteria and fungi. However, in people with tinea pedis, the diversity of bacteria and fungi species was shown to be lower in tinea pedis patients than healthy controls. Double the trouble, Trichophytum rubrum has these two co-workers, Staphylococcus aureus and A. streptococcus. Staphylococcus aureus and A. streptococcus are commonly found in tinea pedis patients and can coexist with Trichophytum rubrum during dermatophyte infections. This can complicate tinea pedis symptoms because the ulcers or exposed skin can cause secondary bacterial infections.
Hello, my name is Connor Volge, and I am presenting the Ophiocordyceps and Ophiocordyceps unilateralis, or what we call the zombie ant fungus. Where does this fungus come from? The main genus or families that it comes from is Hypocrealis cordyceps, Ophiocordyceps, This can actually be seen by the actual phenological trees that have been done by researchers in this area, to where certain genetic actual sequences have shown that the main actual predominant families or genuses that comprise Ophiocordyceps unilateralis comprise of these three main different types. If you would like to actually look up a little bit more of this, here is an article in the actual presentation that I'm doing that would actually lead you to seeing this. The picture that I'm showing is actually one of the ending stages of one of the ants, and Unilateralis really likes to infect carpenter ants, really specifically. Once actually the ant has been infected or the actual spores have attached to the ant, eventually long tendril-like pincers or almost rods will enter through the exoskeleton of the ant that it's infecting. Specifically, Unilateralis really likes infecting carpenter ants. Will not enter its brain, but enter the actual muscles of the ant and begin to take control of it. Once it's actually taking control of it, the ant will be exhibiting certain behaviors that are not normal to ant-like behavior. It will begin pincing or biting or doing or moving in sporadic ways, but this is not because of brain control or neural control of the ant. This is because of protein repression or genetic repression within the proteins that the fungus is taking control of. What you can see here is actually a ascospore stemming directly from its host ant. What are the well, while this is horrible and some of the qualities of this creature are terrible, or this creature, that this fungus are horrible, what are some of the positives that can come from it? Well, from the article that you can see here, some of the possible actual researched positives of this fungus are cancer suppression, some which stem to reducing the metastasis, or actually reducing the metastasis or the spread of certain cancers, but reducing the size of specific tumors, some of which include breast cancer, but overall tumor growth. Some actually show the ability to inhibit or control HIV or the spread of HIV, which is fantastic. But while that is true of this fungus, what are some of the more quote unquote scary zombie like qualities of it? So, how does this pathogenic fungus work? Because some of its metabolites, including its nucleosides, its polysaccharides, its proteins, show some really great positives in terms of in terms of cancer control and HIV control. What are some of the negatives of it? Well, it seems to be now, just like what we've seen, and possibly if you know the show The Last of Us or the video game The Last of Us, that it has the ability to actually take control of the host. Well, thankfully for us humans, that only seems to be true of insects. It enters the host by suppressing the genes of it. But what genes? Protein genes. The fungus, like I stated earlier, does not affect the brain of the host. The fungus, however, does affect the protein genes of its host. What we're seeing here in this illustration that I'm showing is actually the enumeration, where you see in picture A, where the cells are green, normal. But what you're starting to see is that as time goes on, you're starting to see more of the fungal cells enter the actual protein structures of its host. And then now in C, what you're seeing is just an enumeration completely of it, beginning to take over completely. Thank you, and I hope I haven't scared you too much, but I really do love this microbial, and I hope you do too, and have a great day. Thank you.
Hey, my name is Israel Pacheco. For this project, I was working on Candida albicans. Candida albicans is a yeast species commonly found in various environments, including the human body. It is known to be an opportunistic pathogen that can cause infections under specific circumstances. C. albicans reversible cellular morphogenesis plays a vital role in its own virulence. It can facilitate an invasion of host tissues, evade phagocytes, and disseminate into the bloodstream. These different forms enable Candida albicans to thrive and survive in diverse environments, contributing to its pathogenicity and ability to cause infections in various host tissues. C. albicans is a predominant cause of disease and is known for its ability to change its shape and form. C. albicans exhibits a broad range of growth temperatures, allowing for it to thrive in various environments. This ability is to grow within the human body, contributes to its opportunistic nature and its capacity to cause infections. This knowledge is crucial for researchers and healthcare professionals in developing effective strategies for the prevention and treatment of Candida-related infections. According to the research in the comparative genomics of the fungal pathogens Candida dubliniensis and Candida albicans by Andrew P. Jackson in 2009, it has been described as the most pathogenic yeast species, the most prevalent cause of opportunistic fungal infections in humans, and as associated with high levels of morbidity and mortality, all by Jackson. Infections caused by invasive fungi are significant contributors to illness and death, particularly among individuals with compromised immune systems and those receiving hospital care. According to the interplay between gut bacteria and the year Candida albicans by J. Christian Perez, 2021, in the journal Gut Microbiomes, common risk factors for invasive candidiasis include intensive care unit stay, central venous catheter use, broad spectrum antibiotics, recent abdominal surgery, and immune suppression. And once again, that's by J. Christian Perez, 2021. Patients in the hospital or more specifically in the intensive care unit, are often susceptible to invasive candidiasis due to their critical conditions, weakened immune system, and prolonged exposure to medical interventions. A catheter and abdominal surgery can provide a pathway for Candida species to enter the bloodstream and cause invasive infections. The use of broad-spectrum antibiotics disrupts the natural balance of microorganisms, and it allows Candida to potentially overgrow. Published in Frontiers in Immunology, Candida albicans yeast, pseudohyphal, and hyphal morphogenesis differentially affects immune recognition by Liliana Mukarima in 2017 states that Candida albicans is a human opportunist pathogen that can grow as yeast, pseudohyphae, or true hyphae in vitro and in vivo, depending on environmental conditions. In terms of morphology, C. albicans typically appears as an oval-shaped unicellular organism. Under a microscope, it can be observed as budding yeasts, where daughter cells emerge from the parent cells and gradually detach to form individual yeast cells. This budding process not only contributes to the overall shape and colony formation of C. albicans, but also plays a significant role in its pathogenicity. C. albicans has the ability to transition into filamentous hyphae, elongated structures that can penetrate and invade the surrounding environment. These hyphae are critical for tissue invasion and colonization. And I actually have a few photos or a few images that have been made. So here's yeast, here's the pseudo hyphae, and here is the actual hyphae. So here you see, eventually these small ones will bud off. Here it'll keep elongating, eventually splitting, and here it keeps growing as well. So the yeast are small and oval shaped, and they measure about 2 to 4 micrometers in diameter. When yeast form, they're unicellular and will reproduce through budding. Both the pseudohyphae and yeast are gram-positive microorganisms. The yeast and pseudohyphal forms are known as polymorphic fungus. Encapsulated and diploid C. albicans will form true hyphae. C. albicans, when observed under electron microscope, can be seen to have anionic complexes on the surface of blastoconidia, germ tubes, and hyphae. C. albicans hyphal cells contain surface and anionic radicals, which are associated with a fuzzy coat that appears external to the wall and seems to play a role in pathogenicity, phagocytosis, and adherence, according to Jose Ruiz Herrera, who we had just brought up earlier. These anionic compounds then could correspond to hydrophobic properties, which are important for virulence. Hydrophobic cells typically grow in greater amounts than hydrophilic cells. Lactobacillus is able to hinder the attachment of C. albicans and its hyphal cells, the hyphal cells anionic radicals to surfaces by producing biosurfactants, which act as physical barriers. This thereby diminishes the ability of the fungus C. albicans to be able to firmly attach and establish colonization. The outer layer of the cell wall is composed mostly of nanoproteins, which form a dense network that provides a structural integrity and facilitates adhesion to host tissues. Beneath this layer lies a polysaccharide-rich matrix, which contains beta-glucans and chitin, which enhances the microorganism's adherence and colonization capabilities. In viewing a medial cell section of the cell, it would be noted that the electron transparent inner layer of the wall is made mostly of polysaccharides, which are the B-glucans and chitin, and small amounts of proteins, according to Jose Ruiz Herrera, with an electron-dense outer layer and assortment of covalent linkages. So once again, the outside, it's covered by a electron-dense outer layer in that cell wall that is built mostly of different types of nanoproteins, which are anchored by either non-covalent bonds or by an assortment of covalent linkages, mainly to short chains of the beta-1 and the 6-glucan and chitin via the beta-1 and the 6-glucan or directly to chitin. According to Molecular Epidemiology, Phylogeny, and Evolution of Candida albicans by Brenda McManus and David Coleman in 2014, Candida species are typically harmless eukaryotic commensal yeasts that are members of the phylum Ascomycota. Once again, according to McManus and Coleman, eukaryotic cells are characterized by a distinct nucleus that holds the genetic material, the DNA and chromosomes. This nucleus is enclosed by a nuclear membrane separating it from the cytoplasm. Additionally, the eukaryotic cells possess membrane-bound organelles, including mitochondria, the endoplasmic reticulum, Golgi apparatus, and lysosomes, which carry out specific functions within the cell. The phylum Ascomycota is a diverse group of fungi known as sac fungi. They're characterized by the presence of asci, which contains ascospores for sexual reproduction. Relatives to C. albicans include Candida glabrata, Candida tropicalis, Candida parapsilosis, and Candida dubliniensis, with C. albicans and C. dubliniensis being the closest relatives. This microbe has an extreme capacity for adaptation, which may be due to evolutionary trauma the fungus previously experienced, which led to various allelic variations in some genes. The progress in attempting to understand its attachment and adherence is significant, as it will help devise better strategies and techniques to manage and mitigate the impact of this fungus. This will ultimately lead to improved outcomes for patients. C. albicans. Sorry, I lost myself. Let's see. So yeah, C. albicans possesses a cell wall with various components that contribute to its virulency. According to FEMS yeast research, FEMS yeast research found in molecular organization of the cell wall of Candida albicans and its relation to pathogenicity, according to Jose Ruiz, that the cell wall of C. albicans is a structure that, 1, comes first into contact with host cells, 2, carries important antigenic determinants of the fungus, 3, is responsible for the adherence of the pathogen, and 4, it establishes a cross-talk with the host. Once again, according to Jose Ruiz Herrera. It's important to highlight the growing knowledge regarding the role of the cell wall components in the disease-causing behavior of C. albicans. It would allow for us to have a better understanding of the fundamental molecular structures that contribute to the fungus' ability to invade and cause harm, and the possibilities for developing more effective approaches to control and combat C. albicans infections is possible. Candida albicans, like other microorganisms, relies on specific nutritional requirements for its growth and metabolism. Candida albicans is a heterotrophic microorganism that obtains its energy from organic sources. It requires carbohydrates, amino acids, lipids, vitamins, and minerals for its growth. Let's see, there's Catarzina and Magdalena and an Encyclopedia of Mycology in 2021 stated that Candida albicans is able to absorb nutrients in the form of glucose and metabolism glycolysis. In the event of entry into the phagocytic cells of the host immune system, the metabolic pathway is switched to glucogenesis. So once again, they stated that Candida albicans is able to absorb nutrients in the form of glucose and metabolism glycolysis. In the event of entry into the phagocytic cells of the host immune system, the metabolic pathway is switched to glucogenesis. And glucogenesis is the process of synthesizing glucose from non-carbohydrate sources. Glucogenesis helps maintain blood glucose levels, synthesizing glucose from non-carbohydrate sources. Glucogenesis helps maintain blood levels and provide energy to vital organs when dietary carbohydrates are scarce. Glucose serves as its preferred carbon source of Candida albicans, while it can also metabolize other sugars, amino acids, fatty acids, and organic acids. Metabolically, Candida albicans is a facultative anaerobe, meaning it can generate energy through both aerobic respiration and fermentation. In the presence of oxygen, Candida albicans undergoes aerobic respiration to efficiently produce energy. However, under anaerobic conditions, it can switch to fermentation to meet its energy requirements. According to the Journal of Fungi's Metabolic Plasticity of Candida albicans in response to the different environmental conditions by Mariana Gallo, 2022, it states in states of hypoxia where oxygen is not available in sufficient amounts at the tissue level to maintain adequate homeostasis. In water, they quoted this, in water, the main objective of the cells is to remain viable. They activate all internal mechanisms necessary to cope with energy needs and to counter environmental stress, such as osmotic stress. Once again, according to Mariana Gallo. And when glucose ends, C. albicans expresses its metabolic plasticity by switching to alternative metabolic pathways, the most effective being alanine, aspartate, glutamate, and metabolism. 2, there's the arginine and proline metabolism, and 3, there's the glycerolipid metabolism, once again according to Mariana Gallo. In studying the conditions that C. albicans is placed in and expressions of specific genes that then activate once the conditions have been triggered through its signaling pathways, it has been noted that the different states, whether it be budding yeasts or hyphal growth, is due to environmental influences. This metabolic flexibility allows Candida albicans to survive and proliferate in various host environments, even those with fluctuating oxygen levels, such as states in hypoxia. The metabolic pathways of Candida albicans plays a vital role in the adaptation and pathogenesis, influencing its ability to cause infections and resist antimicrobial treatments. Under specific conditions, Candida albicans and Escherichia coli, despite their pathogenic nature, they can engage in cooperative interactions. They exchange nutrients in polymicrobial environments, such as biofilms, with Candida albicans providing structural support for E. coli, while E. coli produces metabolites that serve as nutrient sources for Candida albicans. And according to an article in Pelos, pathogens, Candida albicans interactions with bacteria in the context of human and disease, which was by Morales and Hogan in 2010. A significant portion of human microbial infections are biofilm associated, wherein the formation of a mixed species biofilm, so in this case E. coli, Candida albicans, could create a protected environment that allows for survival to external assaults and also facilitates different bacterial fungal interactions. Once again, that was Morales and Hogan. Bacterial and fungal stimulations or bacterial and fungal simultaneous infections have been linked to increased host colonization and virulence. For example, Candida albicans and E. coli, they display a collaborative relationship in which E. coli boosts the attachment of C. albicans to the mucous membrane of the bladder, which thereby elevates the chances of fungal urinary tract infections. There was also Streptococcus gordoni, which is another microbacteria which promotes C. albicans by secreting metabolic products that C. albicans then uses as a carbon source. As C. albicans grows, it provides a bacterial growth environment with favorable conditions for S. gordoni. The conditions have been created that will allow for the formation of mature fungal bacterial biofilms surrounded by an extracellular matrix to which other bacterial or fungal species can then bind. And that was by Morales and Hogan once again. As more bacterial or fungal species are now able to bind to these infected sites in the oral cavity, it would become more difficult to create treatment options for a growing issue. And while C. albicans is able to work with various microorganisms to further damage its host, other microorganisms are still trying to outcompete for adhesion to the host. So, for example, lactobacillus in its normal inhabitation of the female reproductive tract would be protective against C. albicans, according to Soledad, protective against C. albicans. And according to Soledad Boris and Covadonga Barbez in their article or in their publishing called Role Played by Lactobacilli in Controlling the Population of Vaginal Pathogens in Volume 2, Issue 5 in 2000, in the year 2000 it was posted. They said that Lactobacilli presents two mechanisms to interfere with pathogens. One, the adherence to the mucus, forming a barrier which prevents colonization by pathogens or competition for the receptors of the epithelial cells. Two, the production of antimicrobial compounds such as hydrogen peroxide, lactic acid, the bacteriocin-like substances, and possibly biosurfactants. And once again, that was by Soledad Boris and Barbez in 2000. And in doing so, lactobacillus is decreasing the adherence of C. albicans to epithelial cells either by outcompeting for attachment sites or by releasing biosurfactants that will physically diminish fungal binding. The hydrogen peroxide, lactic acid, or other fatty acids being released by lactobacillus hinders the production and formation of the invasive hyphae. So overall, to come back, Candida albicans is a yeast species which is commonly found in diverse environments, including the human body, and it possesses a polymorphic nature that contributes to its pathogenicity and ability to cause infections. The reversible cellular morphogenesis of C. albicans that plays a very important role in facilitating tissue invasion, evading phagocytes, and disseminating into the bloodstream. Its capacity to thrive and survive in various host tissues is supported by the broad range of growth temperatures it can adapt to. C. albicans is considered the primary cause of opportunistic fungal infections in humans associated with significant morbidity and mortality rates. The pathogenesis of C. albicans infections is influenced by its interactions with other microorganisms and its metabolic plasticity. Cooperative interactions with bacteria such as Escherichia coli or E. coli and Streptococcus gordoni in mixed species biofilms can enhance colonization and virulence. Conversely, certain microorganisms like lactobacillus exert protective effects against C. albicans through mechanisms such as competition for binding sites and the production of antimicrobial compounds. So understanding the complex molecular factors, cell wall composition, metabolic pathways, and microbial interactions of C. albicans is crucial for the development of effective preventative and therapeutic approaches against Candida-related infections. And so lastly, I have some drawings just to be able to show off what this looks like and how it starts off. So here you can go from just one, right? You're at your basic unit, and eventually if it's turning into pseudo-hyphae, or it's just staying as yeast, it'll keep growing like this. Once again, you can see it here. This is what it's supposed to be like if you were to look at it under a microscope. So here you would have the yeast, you would have the pseudohyphae, and you'd have the hyphae. We also have how you would see it. Once again here, you would have it starting off as a yeast, but it can also change to pseudohyphae. And what we forgot to include was also just the other hyphae that I can switch to. So it's not just yeast to pseudohyphae. It can also be pseudohyphae to hyphae to yeast. And these are the different ways that it would look going against different tissues. And so here you see it's moving through these different tissues. This is a mix of hyphae and pseudo-hyphae here. This is just the commencement, really. Here you see the yeast, and they're starting to duplicate. Eventually, they'll split off. They're just growing on top of this tissue. And eventually, here you see all of it. You see the yeast, you see the hyphae, and you see the pseudo-hyphae just disseminating throughout, just really growing against the tissue, okay? And for final drawing, here you are also able to see it. You see the host cells. You see the adhesion to these host cells. You can see its invasion when it's causing damage to tissue, eventually growing in. And here it is. Zygomotropism. Here you can see a biofilm formation that is growing on top of a catheter. Pretend this is a catheter. And also, none of these are supposed to show up yellow, just the artistic rendering of it, or artistic version of it growing on a catheter. It's created its biofilm, will continue growing. And you just have its dimorphism as well. This is what it can grow to. It can grow to the hyphae, or it can grow to the pseudo-hyphae. So I hope you learned a lot. And that, once again, is Candida albicans. My name is Israel Pacheco. That was for Portland Community College, Bio 234. I think spring 2023. Thank you.
Hello, I'm going to be talking about Geogemma barrasi. This archaea holds the record for the ability to survive in the hottest temperature. This organism was first discovered in the Pacific Ocean off the coast of Washington State. On the edge of Juan de Fuca tectonic plate sits a 500-meter-long maze of hydrothermal vents known as Mothra Field. Mothra is made up of 6 clusters of sulfide towers. Geogemma barrasii was found in the cluster known as Faulty Towers, specifically on Fin Tower. Here you can see the Faulty Tower cluster with Fin labeled in the back there. These towers are deep-sea hydrothermal vents that sit on the ocean floor and can reach 24 meters tall. They emit dark plumes of fluid that can be up to 320 degrees Celsius because it's heated by the Earth's magma. Even though this is above boiling, the high pressure of the deep sea keeps it from vaporizing. The towers themselves are mostly made up of sulfide. Magnesium deposits and iron-rich substrate can also be found throughout the field. Despite extreme environment, there's a surprising amount of microbial activity and even some macrofauna. These images are taken on different towers depicting some of that life. G. barrasi is about 1 micrometer in diameter, coccus-shaped, and has a single-layer cell envelope depicted in that close-up. For outer membrane structures, it has 1 bundle of flagella at the end. The bundle isn't visible in this image because this is a small slice of the organism. In this image, you can see that bundle of flagella on the bottom right of the cell. Part of what makes this organism remarkable is its somewhat unique form of respiration. It is a chemolithoautotroph and converts ferric iron into ferrous iron using ferric iron as its final electron acceptor. As a result, it produces a magnetite byproduct. The researchers that discovered strain 121, Kashefi and Lovely, postulate that this actually may have been the first form of respiration, and I'll get into that a bit later. This microbe is also named strain 121 because of its ability to survive and even thrive at that temperature. It grows between 85 and 121 degrees Celsius. Outside that range, cells will stop dividing. Strain 121 can actually survive temperatures up to 130 degrees for 2 hours, and the organism will continue to grow after being moved back to a cooler medium. This temperature is significant because this is what we autoclave at. Up until this discovery, it was thought that nothing could survive, but strain 121 actually doubled in cell numbers after a 24-hour incubation in an autoclave. Here's the phylogeny for G. barrasi. There are 2 other known species of Geogemma also found in deep-sea hydrothermal vents. They are similarly hypothermophilic, though they cannot withstand quite as high of temperatures as strain 121. They also use ferric iron as their final electron acceptors, and not much is known about strain 121's genome other than it having a circular chromosome and sharing over 95% of its DNA with 2 organisms, Pyrodictium occulum and Pyrobaculum aerophyllum. There are a few reasons why researchers are interested in Geogemma's evolutionary role. It is a member of the tax superphylum, and members of this group share some unique characteristics with eukaryotic cells. These kinds of relationships can tell us more about our evolutionary history. When Earth was in its infancy, it was both very hot and very rich in iron. Geogemma are distinctly found in iron-rich hydrothermal vents, which are the deepest, hottest parts of our biosphere. This is why Kashefi and Lovely postulate that this may have been a very early form of respiration. Geogemma boracii's discovery was scientifically significant in a few ways. As I mentioned before, it is the first organism known to survive and even thrive in autoclave, defying what we had previously thought was a rule. This leads into the question of how hot is too hot. The researchers who made this discovery suggest that 150 degrees may be the upper limit there, because past that temperature, macromolecules will start to break down in fractions of seconds, so they wouldn't really be able to support life. That said, we are constantly making new discoveries that defy what we thought were limiting factors. This kind of research is also really important in looking for life on other planets. Finding organisms in these extreme environments makes it more plausible that an organism could survive somewhere we previously considered to be uninhabitable. This is my art component to this project. At the bottom is a drawing of faulty towers where Geogemma boracii was found. The top left is a drawing of the organism with its flagella. On the top right is that cross-section drawing surrounded by magnetite, and the threads represent the black plumes of fluid that the smoker towers emit.
Hello everybody, today I would like to present a comprehensive overview of Plasmodium falciparum, the deadly microorganism responsible for causing malaria in humans. Plasmodium falciparum is a protozoan parasite. It is the most deadly form of malaria and kills about 400,000 people each year, mostly children under the age of 5. The parasite is most common in tropical and subtropical regions, but it can be found in other parts of the world as well. Plasmodium falciparum is a single-celled organism that is about 5 to 10 micrometers in diameter. It has different organelles, including a nucleus, a single mitochondrion, an endoplasmic reticulum, and a rudimentary Golgi apparatus. Plasmodium falciparum belongs to the Epicomplexia phylum. Plasmodium falciparum is closely related to other species of Plasmodium that causes malaria in humans as well. Plasmodium vivax and Plasmodium ovale, and many more. Plasmodium falciparum is an obligate parasite that obtains its nutrients from the host cell, and its life cycle is complex and involves a number of different stages. The first stage involves a female Anopheles mosquito, which will bite or inject sporozoites into hosts. In the second stage, the sporozoites in blood will move to the liver, and in the liver, the hepatocytes infected by sporozoites that mature to chitosan that ruptures and releases merozoites, and in the fourth stage, the released merozoites infect erythrocytes or the red blood cells inside the host body. In the fifth stage, the gametocytes matures in infected erythrocytes, the red blood cells, and are released into the bloodstream. In the sixth stage, the mosquito ingests gametocytes onto the male and female from blood of infected hosts. And in the last stage, the cycle repeats itself with a female Anopheles mosquito biting into the host. Plasmodium falciparum has a profound pathological impact, such as causing severe illness and mortality, particularly among young children. The symptoms of malaria include fever, chills, and sweating, which can occur in cycles. Symptoms may include headache, muscle and joint pain, fatigue, nausea, and vomiting. In more severe cases, malaria can lead to complications such as anemia, jaundice, organ failure, and even coma or death. In conclusion, Plasmodium falciparum is a serious public health issue that causes malaria in humans. It is the most deadly form of malaria and kills about 400,000 people every year. There are a number of things that can be done to prevent and control Plasmodium falciparum malaria, including improving access to clean water and sanitation, using insect repellent, sleeping under mosquito nets, and taking anti-malarial drugs. With concerted efforts, it is possible to reduce the burden of Plasmodium falciparum malaria and save lives. I hope this presentation has been informative. Thank you for your time and have a great day. Thank you.